Thank you, and thanks to the organizers for inviting me. Always a pleasure. And having looked at the weather report from DC this morning, when I got up, it was 18 with a wind chill of 2. It is very grateful to be in San Francisco at the moment. So what I'm going to do is I'm going to go over a lot of the different opportunities and programs that we have that we've established at NIAID to enable activities within the infectious disease space. We've recognized that this has been an evolving situation of antimicrobial resistance becoming a more dominant issue. At the same time, for a lot of different reasons, none of which is really solvable by a single fix. The pharmaceutical companies in general have de-emphasized antimicrobial and in general anti-infective product development. And just a change in the way pharmaceutical development is done, we recognize that we needed to move things further than just supporting scientific breakthroughs. And so I'm going to try to go through what we've done and what we can offer the biotech community and the pharmaceutical community and the academic community in terms of advancing antimicrobial activities. So I think we need to recognize that 50 years ago we really thought we had taken infectious diseases off the table. Many people are not aware that there was active discussion within the medical community of actually doing away with the whole subspecialty of infectious diseases, with the assumption that there could just be a few subject matter experts who could revise annually a bug-drug combination book that all clinicians would be able to follow. This is coupled with the fact that as technology has improved, we've actually recognized there's a lot more infectious diseases, and this is just a snapshot of emerging and re-emerging infectious diseases around the world. We're much better at finding them. We've identified them much quicker. We can characterize them much more rapidly. And so we now recognize that there's a lot of things out there, and there's a lot more things than we had really appreciated. This paper is becoming a little dated, but this was an attempt to try to quantitate that. And in the period between 1940 and 2004, there were 335 new infectious diseases that were recognized. And so this is an ongoing issue. We don't think this is going to abate. There's a lot of reasons to assume that we will continue to see an evolution in the emergence and reemergence of infectious diseases and the presentation of infectious diseases in different situations. I think the West African Ebola outbreak is instructive for a number of reasons, but one of which I don't think has been truly appreciated, is that while we saw isolated sporadic outbreaks in low population density events occurring for 40 years, in the West African outbreak we saw what can really happen when there's inadequate public health infrastructure on top of a setting of reasonably good road systems to let people migrate from one area to another, and you saw a very rapid progression of disease that was totally unexpected. So I think we also need to recognize that we're in the 21st century, and a lot of the ways we approached infectious diseases historically just don't seem to make as much sense anymore. Always taking lots and lots of pills may not always be best. Sharp ends of needles are not always very enjoyed by people. And the way flu vaccines are still made to a large extent, it really is more of seems like an agricultural enterprise than a pharmaceutical enterprise. So at NIAID we have a number of focus and capabilities that I want to go through briefly. Historically, and as most people recognize, the bread and butter of NIH is basic research, but part of this in infectious diseases is really a focus on the discovery and concept identification, the concepts that can lead to interventions to interfere or intervene in an infectious disease process, to terminate it. One of the big areas that we saw that was really lost as the pharmaceutical industry abandoned infectious diseases in general was early stage translational support. It's not easy, it's not trivial, it takes a lot of different skill sets to do, And the academic community is not necessarily always well positioned to do that. In biotech companies, while this is what they're trying to do, limitations both in terms of resources and intellectual capacity, just because of the numbers of people involved, really makes it a difficult task. Product development capabilities, you get into really taking a product and moving it forward. There's a lot of expertise that's involved, things that are not very sexy, not exciting to do, manufacturing, regulatory, the kinds of things that make people's eyes glaze over. And then one thing that we've begun to recognize in recent years is that if you really want to have novel interventional concepts, the problem that comes with that is the regulatory oversight is always behind the curve. There's always questions, there's always issues, there's always potential areas that need to be addressed when you're talking about new modes of intervening. There's always concerns. Demonstration of safety and efficacy are always paramount. And with new ways of doing things, there's always questions underlying the methodology that you are using to demonstrate that safety and efficacy. And so we've recognized that part of our role, because it's difficult for any single company to do this or single entity to do this, is working in partnership with the FDA to try to support the necessary regulatory science to enable these novel products to actually move forward. So there's enough of a fundamental base of knowledge and information that the FDA can draw on to give them a particular comfort level in order to move forward. So we've talked about the idea of broad spectrum approaches. I think most people, when they think of broad spectrum activity. They think of antibiotics that hit a lot of different types of bugs. We're even more expansive than that, of medical countermeasures that are applicable to classes of threats, not just one particular class. We've looked in recent years, because of the science leading us in this direction, to host-directed interventions. We normally think about anti-infective therapy of killing a bug, and the reality is that you don't get a product approved by the FDA that kills a bug. You get a product approved that prevents disease. And so if you can actually interfere with a host process that the infecting organism requires in order to mediate disease, or is required by the organism in order to complete its replicative cycle, you can have as much of an impact. And this has been done we have some examples of this in already approved drugs. One nice example I always like to cite is that in the case of tetanus, we have a tetanus vaccine. That vaccine does not kill the organism. That does not prevent anything about the organism itself in terms of its replicative life cycle. It actually just prevents the toxin from mediating disease. And so if you eliminate the toxin-mediated disease aspect, the rest of the infectious process is handled without much fanfare. And we're never talking about drug resistance to Clostridium tetani. Immunomodulators are another area that we've seen a large emerging science. This came out of largely trying to develop better adjuvants for vaccines, but many of these, in fact, have some standalone properties and may either be standalone agents in and of themselves or potentially adjuncts in combination with other traditional anti-infective products. And then we see this most commonly in the flu area of universal or strain-independent immunity, this concept of exactly moving beyond a sort of simple virus neutralization type activity from an antibody to other immunological mechanisms is gaining more and more attention as human immunology expands. But beyond the activities themselves, there are technological broad-spectrum advances, temperature stabilization, alternative delivery devices, and vaccine adjuvants that have broad applicability across a wide range of product categories. And then finally, in the area of platforms, both in terms of production and diagnostics, broad spectrum I think is very clear. Standardized manufacturing systems. The reason we can make a flu vaccine new every year to address strain drift is because of a standardized manufacturing system. We don't have to go back and re-engineer the manufacturing process. In the diagnostic realm, we've seen slowly over time the introduction of multiplex diagnostic systems. So rather than just looking for an agent, looking for the spectrum of agents that can cause that particular, is involved in a particular condition under investigation. And then beyond that, we're actually looking at the possibility of agent-independent diagnostic systems. This is really looking at the possibility of using host evaluation. Again, people will sometimes find this to be a little fanciful, but I like to remind people that fever and an elevated white count is a pretty good indicator of infection. And so what I'm looking for here is something a little more sophisticated than that. So a couple of, just one slide on terminology, because a lot of words get bandied about. This is the language that I use and how we've sort of structured what you'll see some of our funding opportunities around. So I like to think of general in terms of product development falling into three general buckets: the basic, translational, and product development. So basic, everyone understands, fundamental science. I'm not looking for a product. Basic is sort of, I don't know what I'm doing. Translational is when you've really identified a concept that you think might have the potential to lead to an interventional product. And that requires a lot of effort in order to demonstrate that that is in fact reducible to practice and that you can actually implement that in a practical, cost-effective manner. And then finally, when you have a product in mind, the product development, the testing and evaluation. Now we shift this into an early and a late, where I usually distinguish around phase 2. The late stage stuff, post phase 2, is clearly all commercial development activities. It's really working on that manufacturing, regulatory, those pivotal safety and efficacy trials. And you'll see this on the next slide why I'll distinguish this. So we put together a series of overlapping funding mechanisms to try to cover these different categories, because quite frankly, if you're talking about wanting funding support for a GLP tox study and formulation study, it's certainly never going to be as sexy as a basic science grant application wanting to identify and characterize the function of a new enzyme or something like that. So we've tried to separate these into different funding categories so that we really are comparing apples with apples. And so the grant portfolio largely is basic with some extension into the translational space. Historically, NIH has done, and I'll have some slides on this later on the small business program, the SBIR program. We're obligated by law to have that program. But we wanted to beef it up a little more, and what we've created is a different category that we refer to as partnerships and centers, and I'll talk a little bit more about that. But they are really the large workhorse for our translational effort, where we feel that there's quite a bit of needed effort to advance a lot of promising scientific discoveries into potential product categories. And finally, when we're really working on a product and trying to advance that product, we work through contract mechanisms. And that's the federal acquisition process. It's very cumbersome, and I know it's difficult for small companies that are forced to play in the same league with large companies who have large legal people, large staffs of legal people who can read excessively boring and dry language on how to respond to this. But I don't apologize because I had nothing to do with it, and I dread it as much as you do. Our typical goal here is to try to move something potentially through phase 1 and into phase 2, and then have a transition partner. And I think it's very important that in the contract mode, we're really looking for something that has a long-term strategy, and that means a transition partner who's going to take it on. It may be a large pharmaceutical company, it may be the company itself, it may be a nonprofit, Gates or other funding agency. If it's a pure biodefense play, it may be BARDA. So we have a number of different types of transition partners that we look for, and we try to structure the program in order to give them what they need in order to move the programs forward. So our centers program is originally we had the regional centers of excellence. We had 11 of these. This was done they had a lifespan of a little over a decade. They were very successful in terms of moving forward in a number of areas that were just simply not well investigated, and they expanded a lot of work and area. We morphed those 2 years ago into the Centers of Excellence for Translational Research to focus specifically on the translational space. And this is the current centers. I put them up there because they're doing a wide array of activities that many companies may in fact have an interest in engaging with them because of unique opportunities. A few I'll point out. Erica Sapphire at Scripps with the Viral Hemorrhagic Fever Consortium actually was working with ZMAP, that was the ZMAP, the MAP Pharmaceuticals, and with the ZMAP product for Ebola. And Tom Geisbart at UTMB has worked on a number of Ebola vaccines for multiple companies, as well as the Tecmira product that was tested in West Africa. There's a wide variety of antibacterial, antiviral. Some people are doing host-directed interventions. There's some diagnostic work that's ongoing. And there's some vaccine platform technology work. All of them have regulatory science components involved to try to support the regulatory science that gets done, and so it's a good area to identify subject matter experts. Now, in the partnership program, we've been doing this for about a decade and a half, and this is some of our more recent ones. They can sometimes be very broad, covering categories A through C, maybe specific just in terms of therapeutics and diagnostics, or in general. And then we've done a number of very targeted ones, novel therapeutics for select anaerobic protozoa, which we felt there was scientific opportunity and insufficient attention being given to those. So these, and you'll see that we have some specifically for diagnostics, vaccines, and therapeutics. And we use two major mechanisms, the R01 and the R21-R33. The R21-R33 is a newer mechanism, and this allows us to entertain a little higher risk because it allows us to fund for the initial 2 years a larger number of candidates, and then to down select after 2 years the most successful ones to receive a larger portion of the pie, because the annual amount of money that goes into this program is held constant over 5 years, but there's more spread out over, there's fewer dollars spread, there's the same amount of dollars but spread out over more awardees in the early years and fewer awardees in the later years. So it allows for higher risk to be worked in. The R01s are more traditional in terms of preclinical candidates. Again, they focus on multidisciplinary, they're performance-based, and generally these can take you all the way up into an IND submission to the FDA. It's been a very successful program, and the information in terms of this can be found on our website. There's the link that you'll have. The SBIR program, I think, is important to emphasize because it's been around a long, long time. I know that people have variability in terms of success with it. One thing I like to always emphasize to people is that there are cycles with this. Because of the different, in terms of payouts between phase 1 and phase 2, what you can have is certain years there's just a lot less money available for phase 1s because there's a lot of phase 2s that are getting awarded. And so if you aren't, it doesn't necessarily mean that the same number of awardees for phase 1 or phase 2s will hold from year to year. There does tend to be cycles. NIAID has applied for waivers so that we can grant a little more money in a number of situations. And there's an individual in our group that specifically deals with this and can provide you with a lot of the details on how this. The biggest one, I think, is that this was something that was an issue that we routinely complained to congressional staffers about, was that venture capital-backed firms were precluded, and that has now been modified, and so the venture capital-backed funding does not preclude you from small business awards, and that's something that needs to be mentioned. We list a number of high priority areas in terms of what we're looking for. They pretty much cover, I'm sure almost anyone working in the infectious disease space can find some aspect of these that covers what their particular area of interest is. And I want to highlight this because, again, this may be old news, but it may be so old that people have forgotten it. But going back to about 2010, Congress actually increased the SBIR set-aside. So historically, the SBIR was at 2.5% of our budget, but this is actually growing and still growing and will max out next year at 3.2% of our total budget. So the funds for this have been slowly increasing, and you can see across NIH, and particularly for NIAID, that it's a quite substantial sum of money to pay out. The STTRs as well, which I think is also a sometimes underutilized source of funding, particularly for technologies that a company is working with that may have additional applicability that an academic can help you work on. That's where the STTR really shines. So those are funding mechanisms where money goes out to you as the investigator, but we've recognized that that's not always sufficient. It's not always timely in particular, and in dealing with public health outbreaks, we have to have mechanisms to be able to respond more quickly. We also recognize that there are particular funding and resource constraints that companies face, and we want to try to help get over that the funding opportunities may not necessarily address in either a timely or comprehensive manner. And so what we've evolved over the last 10 years is a series of services that we offer to the scientific community that includes both therapeutics, vaccines, and then spans into clinical. And that the key thing here is that this is no cost to you in terms of utilizing these services, but there are no funds to you. So after your lawyers and my lawyers behave no differently from your lawyers in that regard, go through extracting their pound of flesh in terms of the material transfer agreements, we can do work for you through a series of contracts that we maintain, where the data and output from those contracts goes back to you to use as you see fit. It's important to emphasize, and not all legal staffs at companies will recognize this or accept this, but it's important to recognize that we've instituted DECs with all of these contracts. A DEC is a declaration of exceptional circumstances, which basically means that my contractors have waived their buy-dole rights, so they retain no intellectual property on the work they do for you. We actually have to get them to waive that because it's the law that they are being funded by the government. They actually have that right. So they have waived that right to do that. Now, they may generate intellectual property in terms of development of assays or testing procedures for your particular product, and that they can retain, but the intellectual property of your product, they do not retain any of that. So we offer a wide variety of in vitro assessment of antimicrobial activity. This is particularly valuable in terms of things like if you want to have something tested against anthrax, where you not only have to have BSL-3 containment, but the laboratory has to have select agent registration. It's not a trivial thing to do. Even large pharmaceutical companies can't do this all the time. And so we maintain a wide array of services for testing. We have the ability to test against panels of drug-resistant organisms. We put together a lot of things that can support the community for testing of candidate products very easily. The interventional agents covers all small molecules, almost anything you can think to do. When I wrote this contract, the first time I wrote this solicitation back in 2004 and was asked what did I want them to do, I said anything the FDA might require for in an IND package submission, and that's basically what they were able to provide us with. The biopharmaceutical products is specifically directed towards biologicals. We know that there's a lot of specialized types of manufacturing issues there, and they can pretty much cover anything, mostly to do with monoclonals, phage, things like that. For vaccines, we have both a testing and a manufacturing component, and this was used extensively. For example, in terms of, there's about a dozen Ebola vaccines that have actually gone into humans, and of those dozen, about 75% of them have actually gone through our specific testing to provide companies with the information they needed in order to move their products forward. And then we have clinical capabilities. We have a phase 1 unit, and we have a collection of vaccine treatment and evaluation units that can test vaccines and therapeutics. And again, that's mostly phase 1, phase 2 type studies. Finally, we have a wide array of animal models that support a lot of the work up above, but also are developing animal models in and of themselves. So particularly for those types of products that may require an animal rule licensure, it's these contracts that are actually developing the pivotal animal efficacy studies that the FDA will be basing decisions on, and we can do those studies to support your products. Oh, there we go. I also want to, one of the other things that we have, and this is just one example, I like it because it has, I think, the best name across government. In terms of extended partnerships, this is the FANG, the Philo, Animal, and Non-Clinical Group. The idea here was we recognized about 6 years ago that the ability to bring filovirus countermeasures forward was difficult because there were few labs working on it and each labs were doing their own thing, and so there needed to be some standardization. And so the FANG was a group that initially was DoD and HHS only to look at regulatory strategies for moving both vaccines and therapeutics for philos forward and how we would do this. This has evolved to where most of the major BSL-4 labs working on philos are now part of this, and there's a number of different working groups. They've had workshops, they've published papers on this, but they have produced standardized reagents, standardized assays that everyone is now using, and it has actually brought together the philo community in a way that wasn't feasible or possible before, and is really the basis, will be the basis for what most of the filovirus countermeasures will be licensed. So I just wanted to hit on a few things in terms of where do we think the field may be going. So I wanted to talk about some next generation vaccine concepts. One is, interestingly enough, I think that some companies are beginning to think about now is passive vaccination. So we've recognized that a major source of morbidity and in some cases mortality for infectious diseases is that many people don't react well or respond well to vaccines. And we put a lot of effort into trying to do this by basically doing more of the same. So we now have a fourfold concentrated flu vaccine for the elderly is one, and the work on adjuvants trying to make stronger and stronger vaccines to boost responses is the way a lot of this has been attacked. Passive vaccination is basically, rather than trying to induce an immune response, is to give somebody an immune response. So the idea, particularly now that we have the capability to mutate antibodies to give them longer half-lives, the possibility is that a monoclonal injection of an antibody may provide a time-limited protection to an individual that would otherwise not be able to mount an effective immune response. And so there's a lot of interest in this now because for certain populations, nursing homes, this may be a preferred way, could be potentially a preferred way to actually protect people against seasonal types of infections, as well as be able to quickly respond to emerging infectious diseases. The second one is one that is just beginning to be talked about, a PII, a prophylactic immune intervention. And this arises from the issue that vaccines suffer from basically having two hurdles from a regulatory perspective. You first have to get through the FDA, which everyone recognizes and everyone accepts, and it's a tough hurdle, but then you make it. But then after that, that isn't enough. You now have to get through another hurdle, and that hurdle is the ACIP, which is the Advisory Council on Immunization Practices, that is basically giving the recommendation that this vaccine should be used, and that is really the gatekeeper in terms of what gets reimbursed by insurance companies. And so you have to get that ACIP recommendation, or insurance companies are not going to pay for it. So the issue is, can we change the name? Can we reframe it and not call it a vaccine so it doesn't go through ACIP? So a prophylactic immune intervention may be, for example, What if I had a vaccine for MRSA? Who would I give that to? We think of MRSA as a big problem, but who's the population you really want to vaccinate? Well, one population you may want to vaccinate is people going in for elective joint replacement surgery, because they're at risk, and if they develop a staph infection in that joint replacement, they run into a lot of problems. You're not really giving that as a vaccine, you're giving that as a prophylactic immune intervention that you can select 1 to 2 months before their elective surgery. You can give them a vaccination to prevent a downstream event, and it has a lot of differences from vaccine because you're not necessarily focused on the duration of immunity in that point. You want to get them through that specific event where they're at a high risk. So there may be a number of examples where we can actually apply this in terms of protecting people through particularly dangerous periods when they're at high risk for some adverse event due to some other situation they're involved with, typically some elective medical procedure or even unelective medical procedure. And so calling it a PII may actually provide a means of developing things that get you away from being considered a vaccine, but will in fact have a very important role in medical treatment. There we go. And so next generation therapeutic concepts, monoclonal antibodies, we have a lot of activity going on. There are companies that are beginning to look at this. We think that monoclonals offer some very good modes in terms of protection, as I mentioned previously, for passive vaccination, but also for treating people with specific diseases. Targeting virulence factors has been something that's been kicking around since even I was working in industry 20 years ago. And there's been several issues here that have always stood in the way, but I think the situation now with gram-negative resistance and some of these resistance factors that are floating around is really encouraging a greater look. The science is much better, and the idea of being able to treat to really reduce the virulence of the infection that may go as far in terms of modulating disease as actually killing the organism, I think is deserving a good solid look. Phage is another one that is really becoming, I think, coming back to the forefront. It was 100 years ago that phage was discovered. It was initially in the early part of the 20th century, was getting quite a bit of attention. Most people probably don't realize that in France, L'Oreal, the hair color company, was actually began as a pharmaceutical entity, and they were heavily involved in making phage. I think the introduction of penicillin actually drove them more towards the health and beauty side, away from the health and more to the beauty side of the curve there. But phage are getting attention for a number of reasons. And I think they're well deserved. We had a workshop at NIAID this past summer that I think it was someone in either Nature or Science referred to it as a landmark workshop. I don't know that it was that fantastic, but it was interesting to see the diversity of people who are interested in advancing phage. Many people are not aware there are licensed phage products already. The FDA has, in fact, licensed phage. They've come through the Center for Food Safety, not through the Center for Biologics. But there is a general impression of the safety of phage. If any of you choose to have a salad for lunch today, you're probably consuming about 500 million phage in that meal alone. So there's a general feeling that phage are relatively safe. And the other interesting number that I learned from another workshop we held on the microbiome is that, surprisingly enough, 7% of your microbiome is killed every day by phage in your microbiome. The interesting feature with the microbiome is when the sequencing studies were first done, they stripped out and threw away all the phage sequence that they were coming up with because it was so overwhelming it prevented them from looking at the bacterial species that were there. When they've gone back and looked at it, it turns out that in fact the phage are a major component, and there's a continual arms race going on between phage and bacteria in our guts. So there's a focus on looking at phage for a number of medical conditions as well as health conditions that deserve attention. And I think in terms of infectious applications, I don't think anytime soon we'll be injecting phage into people's bloodstreams, but I think topical and oral applications and even urinary tract may have some very good applications where phage can be used. Another area is in the area of decolonization as a way of reducing the usage of antibiotics as a means for decolonization, and that may have a big impact in terms of antimicrobial resistance. Finally, the one we've seen some effort, we've actually been able to advance at least one product into phase 1 testing, is host-based targeting. As I mentioned earlier, the ability to target the host, which again is scary to many people, but I like to remind people that this is what the pharmaceutical industry tries to do every single day, and they're doing it on a regular basis. So targeting the host in order to change the ability of an infectious agent to either replicate efficiently within the host or to manifest disease, I think, is another viable strategy that deserves some attention. So overall at NIAID, what we're trying to do now and into the future, we have to maintain a microbial, we have to maintain a warm base across the microbial spectrum. We'll continue to focus on high consequence threats regardless of source. This is always a situation, whether it's intentional, accidental, or natural, we're always going to be, the high consequence threats are the ones that are going to gain our attention. But again, the focus on broad spectrum agents, both antibacterial and antiviral, because the development of broad spectrum means that potentially an intervention we make for one condition today may in fact be something we have already on the shelf applicable to something new that emerges in the future. I think the universal vaccine concept's being actively pursued for flu. I think we'll see that on other areas as well. Coronavirus has come to mind immediately as another class of agents that would be wonderful to have universal vaccines rather than just waiting for every SARS and MERS to emerge each decade and make a different vaccine for that. Then the use of non-traditional methods to reduce antibiotic usage, I think, will be continue to gain a lot of attention. And then finally, the platform technologies that both enhance our ability to detect and to speed production are the ones that we're really focused on. So I'll stop there and take any questions. Thank you.
So in the speakers, the translation, do they do the vital?
No. No, they don't. They don't. I should emphasize each one of them is oriented around a particular theme, and you can find all the information on them on our website.
Yes. Could you amplify what you mean by the non-traditional?
So two that come to mind immediately, I think, are monoclonals and phage. Come to mind immediately. One that's probably would be pseudo non-traditional is we've employed a number of what we refer to as strategy trials. So this is not identifying a new anti-infective, but using it in a different way. In many instances, for example, in antibiotics, they get approved with a standard treatment course, and clinicians begin to question, well, do I have to give it for 14 days? Could I maybe do 7 or 10, or can I reduce the amount? That's not something that the company necessarily is interested in. It's incredible expense to do. So there are some legitimate questions and issues about how things are being done. Yes, there's also right. There's also more. I don't have a good word for it, but standard things that are done in the hospital that maybe people question. If I did it this way, there would be less. Whether or not changing IV lines, those sorts of things, practices that could maybe be modified.
And do phages, I would imagine they develop a resistance as well, or are they just always going to be on the list?
Well, organisms do become resistant to phage. What's typically done for the in the food safety is that there's a cocktail. So first of all, you employ a cocktail of phages, and there's a monitoring that goes on just in the same way you do with antimicrobial resistance in terms of drug sensitivity that goes on, and occasionally the decision has to be made as to whether you swap out one phage and throw in another one, and that's a regulatory issue that SBIR is not prepared at this time to answer, but that came up at the workshop, is would swapping out phage in a cocktail be considered a strain change, or would you have a whole new product that you would then have to go through and demonstrate? And that they would have to identify some resistance, right? And it's conceivable that, over time microbial populations would shift and either become resistant to phage, or new microbial populations would slowly evolve that are just not sensitive to the phage, but haven't developed specific resistance to the old phage, but they're just new. Yes.
How does the company approach the institution to work with you for the services?
So if you go on our website, which is not exactly the not exactly the most user friendly, but there is a link should be on the front page of the NIAID website, not the NIH website, but once you get into the Institute website, that is called Resources for Researchers. And once you're in that link, you can then drill down through a series of menus. And what was a little bit thoughtful and was very deliberate in how it was done is they were not made to be single path drill downs, in that you can get to the same place many different ways by either talking about, I have an interest in an organism, I have an interest in a activity, I have an interest in a product class, and it'll get you to the same place eventually with the details of that particular service that you're interested in. Every one of those pages will have a specific contact name on there that you can email that person directly in terms of finding out further information on what you need to do and who you can speak with in terms of accessing the service. It's open to anyone. Anyone. Yes. So HHS does have OTA, other transaction authorities, and because of HHS having it, it does flow down to NIH. So theoretically, I have the capability. Now, knowing that I have the capability and someone letting me use that capability is another matter. What I do know is that my contracting official, the guy who runs my contracting shop, and there are a total of 4 contracting shops at NIH total, one of which is just my institute. So we do a major portion of contracting. And he was involved in long-standing and ongoing discussions at HHS into how and when these authorities would the ability to utilize OTA would be used. And BARDA has in fact entered into a few OTA agreements. NIH is still opining on whether they would allow that to be done. And that unfortunately is a discussion that has to occur at a policy level before there's any scientific rationale for why it would be done, and it's still up in the air. I'm not holding my breath for the short term. Okay, thank you.